Good afternoon, children.
Good afternoon, ma'am.
We were discussing about transcription, and after so many days we are meeting now. I don't think you have forgotten. Have you forgotten? Only the introduction part, a transcription unit. We had learned which are the three parts of a transcription unit, Malaviga. Three main parts of a transcription unit.
Promoter, terminator, and a structural gene.
Structural gene. So we are going to learn in detail about the process of transcription. So you know what is meant by a transcription unit, the different components of a transcription unit. Now you need to learn how this transcription process is taking place. We also compared transcription and replication. So transcription is the process by which genetic information present on DNA is getting copied to RNA. And here RNA polymerase is the enzyme needed for carrying out the process. Before we proceed to the process of transcription, let's learn about the different types of RNA. RNA are of different types. In prokaryotes, there are three types of RNAs. You hope you know the difference between prokaryotes and eukaryotes. Bacteria and certain other organisms are coming under prokaryotic organisms, whereas eukaryotes, all the higher plants and animals are included under eukaryotes. The main difference is that in eukaryotes, a nuclear membrane, a well-defined nuclear membrane is present, whereas in prokaryotes, there is no nuclear membrane, only the genetic material, and you call it as nucleoid. Nucleoid is a genetic material present in prokaryotes. They don't contain or they don't have a membrane covering it or separating it from the cytoplasm. And in prokaryotes, there are three major types of RNAs. Messenger RNA, transfer RNA, and ribosomal RNA. These are the three types of RNAs present. Ribosomal RNA, messenger RNA, and transfer RNA. And these three RNAs are necessary for the production of proteins. You know that once transcription process is completed, once RNA is formed, it has to get translated into proteins by a process known as translation. So for translation or for the synthesis of proteins, all these three RNAs are required. Each one is having its own role. Each one is having a specific role. Together, three of them will help in bringing about protein synthesis. And first, let's take the case of messenger RNA. Messenger RNA, picture is given over there. They will provide the template and the genetic information. It is having the genetic information in the form of genetic code. So there in the figure, you can find messenger RNA and you can see some pillar like some block like structures. Those are the different or three of them together form what you call as genetic code, a three-letter code, three of them, three nitrogen bases. So what you see like these pillars are the, not like pillars in the figure it seems to be. So those are the nitrogen bases and a set of three nitrogen bases will form a triplet codon about which you will be learning later. So three of them will form a genetic codon. It is coming in sets of three. A three-letter code or three nitrogen bases together will form a genetic code. So messenger RNA, they will act as a template. And why it is acting as a template or what is its role, or by acting as a template, how it is resulting in the formation of proteins, you will learn later when you are learning about translation. So messenger RNA, they will act as a template for the protein synthesis and it is having genetic information in the form of genetic code. Don't worry about genetic code also. You are going to learn all these in detail. So when I complete the genetic code and the translation process, you will understand it better. Now just understand what functions they are performing. They will act as a template and they contain genetic information in the form of genetic code. Details you will learn. Then transfer RNA, the third picture, transfer RNA, they will bring amino acids and they will read the genetic code of messenger RNA. So here I told there is a triplet codon. Say for example, if it is AUG, and if the transfer RNA AT, okay, just a minute. Okay, so if the messenger RNA, I told this is a single codon, genetic codon, a sequence of 3 nitrogen bases will form a codon. So that one codon, and here you can see the matching codon here for the transfer RNA, UGC, A, adenine, it has to pair with thymine, but since it is RNA, it will be uracil, then C, cytosine to guanine, guanine to cytosine. So the transfer RNA brings amino acids and they will read the genetic code of messenger RNA. So each amino acid is having a particular codon and they will be carrying, they will come and read this. So this transfer RNA will read the codon present on it, and only the transfer RNA which is complementary to that sequence will come and join over there and transfer the amino acids. So when I'm saying like this, you may not understand. Only when I finish explaining the process of translation, you will understand it. So even if you haven't understood it, not to worry. I'll repeat it again. The transfer RNA brings amino acids and reads the genetic code present on messenger RNA. You learn like this now for the time being. So don't worry about the details of the process, that you will understand when we learn about translation. So there are 3 types of RNAs: transfer RNA, messenger RNA, ribosomal RNA. And briefly about their functions. Messenger RNA, they will act as a template, and they contain genetic information in the form of genetic code. Then transfer RNA, transferring. So this transfer RNA will read the information of the genetic code present on messenger RNA, and accordingly they will be transferring the amino acids. Once again, transfer RNA will read the code, genetic code present on messenger RNA and brings the amino acids. And the third one, ribosomal RNA. Ribosomal RNA is a structural part of ribosome. Ribosome you have learned when we had discussed about the different cell organelles, ribosomes, which are the site of protein synthesis. So ribosomal RNA is an important part of ribosome, and they play a catalytic role. They can catalyze the process of translation. They can catalyze the synthesis of protein. So these are the 3 RNAs and their functions. I'll repeat once again. So don't worry about the details of these, which you will be learning gradually. As we proceed, we'll be learning all these. There are 3 types of RNAs present in prokaryotes. And these are transfer RNA, ribosomal RNA, messenger RNA. Briefly about their functions. Messenger RNA, it will act as a template, and they have genetic information in the form of genetic code. Then transfer RNA. So the genetic code present on messenger RNA will be read by transfer RNA, and it will bring proportionate amino acids. The corresponding amino acids will be brought to there. Then ribosomal RNA. Ribosomal RNA or rRNA, ribosomal RNA or rRNA will catalyze the process of translation. The translation process is catalyzed by ribosomal RNA. It is an important part of ribosome. Ribosome is a cell organelle which acts as the site of protein synthesis. Is this clear? Shall I proceed?
Ma'am,
Ah,
from where does this amino acid come?
that we will learn. We'll be learning once we learn about translation. Some of the amino acids are synthesized by your body. Some of the amino acids we have to take in through our diet. Essential and non-essential amino acids, I think you might have learned in lower classes. We have different amino acids and some need to be synthesized by your body. Some will be taken in through the diet. We will be taking it, okay, from the food. Once it is, you know that upon digestion, proteins are broken down into amino acids. Once digestion is completed in the small intestine. Hope you're remembering it. So some of the amino acids we are obtaining from the food we are taking. Process of transcription in prokaryotes. First, we are going to learn about the transcription process taking place in prokaryotes. And here from the picture, a few points you might have understood. That is, this is our DNA, double-stranded DNA. And I have told you only one segment is taking part. Out of the two strands, only one strand is taking part, and that too only a small portion of the strand. See, this part is taking part. That particular part is taking part in the process, and an RNA is being synthesized, or the genetic information present on this part of DNA is copied to RNA, and the synthesized RNA you can find here. And the enzyme which helps is RNA polymerase enzyme. In replication, the enzyme was DNA polymerase, DNA dependent DNA polymerase. Here it is DNA dependent RNA polymerase. What is to be remembered is that the whole DNA strand is not taking part in the process, only a small part, only the transcription unit, only that part will be taking part in the process. And only one of the strand is taking part out of the two strands of DNA, only one strand is taking part, and the information present in this transcription unit or present in that particular region will be copied to RNA, which you call as transcription process. The enzyme is RNA polymerase. There is a single DNA dependent RNA polymerase that catalyzes transcription or synthesis of three types of RNAs in prokaryotes. And when it comes to eukaryotes, there are different enzymes, but in prokaryotes, there is only one enzyme, that is DNA dependent RNA polymerase. Only this enzyme is involved in the synthesis of different types of RNAs and also in the transcription process. The transcription process and synthesis of various RNAs are catalyzed by just one enzyme, that is DNA dependent RNA polymerase in prokaryotes. The RNA polymerase is capable of catalyzing the process of elongation. So the polymerase enzyme will help in catalyzing the elongation. Now, elongation, termination, all that we will learn. Elongation means the process proceeds or the transcription proceeds and we get messenger RNA. So the elongation of that messenger RNA will be, or the RNA will be carried out by RNA polymerase enzyme. And this RNA polymerase enzyme, for the process to begin, the process should start. And already in the previous class we learned in a transcription unit, there is a promoter where the process starts, terminator where the process ends. So for the process to begin, this particular enzyme will associate transiently, a temporary association with a factor known as initiation factor. There are two factors, and this enzyme will get associated with these enzymes to bring about the termination and initiation. That is why in the previous line it is mentioned, this can only catalyze elongation process. It can only catalyze elongation process. But if the process is to start or to end, It needs the assistance of other factors known as initiation factor and termination factor. I'll repeat once again, in prokaryotes, there is just one enzyme which catalyzes the synthesis of different RNAs and also the transcription process. RNA polymerase can only catalyze the elongation procedure, but the starting and ending, the beginning of the transcription process and the termination cannot be initiated by our RNA polymerase. It needs the assistance of certain other factors. For the process to begin, it has to temporarily get associated with the initiation factor, and for the process to end, it has to get associated with another factor known as termination factor. Initiation factor, the symbol is sigma, and for termination factor, the symbol is rho. Rho factor and sigma factor. Hope you understood what I said. Yes? The significance of these two factors, I hope you have understood. Because RNA polymerase, they cannot initiate and terminate the process. They can carry out elongation, but if the process is to start, or if it has to come to an end, it needs the assistance, or it has to get associated with termination factor, that is rho factor, and initiation factor or sigma factor. Now, the process of transcription in prokaryotes, it is completed in 3 steps: initiation, termination, and elongation. As you can see in the figure, initiation, elongation, termination. This is carried out with the help of RNA polymerase enzyme alone, but for this and this process to be carried out, those factors which I mentioned are also necessary. So RNA polymerase enzyme will temporarily associate with those factors to initiate and terminate the process. Initiation. RNA polymerase enzyme binds with the promoter and starts transcription. The promoter of the DNA is recognized by the initiation factor or sigma factor. So RNA polymerase, as you can see in the figure, this is RNA polymerase. This is RNA polymerase. So they will get associated with the sigma factor, and with the help of which they will recognize the promoter. And that is how the promoter is recognized. And they will bind there and the process will start. RNA polymerase will bind to that site known as promoter and the transcription process will start. And this recognition of the promoter site is done with the help of initiation factor. So this RNA polymerase will get associated with the sigma factor. Together they will go and bind with the promoter region, okay, which will start the process. Now next is elongation. Already you can see in the figure, for the process to start, the RNA polymerase and the sigma factor has got attached to the promoter. Now the next,
can you show the previous slide once more?
I will repeat it. The next one is initiation. So I told for the initiation factor, sorry, next one is elongation. So before that, initiation we'll see once again, RNA polymerase has to bind with the promoter. Only then the process will start. But DNA dependent RNA polymerase cannot recognize the promoter and bind to it in the absence of the sigma factor or the initiation factor. So RNA polymerase first will get associated with the sigma factor and then they will go and bind with the promoter. That sigma factor is essential for the recognition of promoter by the enzyme. Otherwise, they will not know where to go and get attached. So the recognition of this particular site, promoter site, is done or is carried out with the help of this sigma factor. And once initiation process is completed, now the next step is elongation. DNA double helix will get unzipped. You can see in the figure the unzipped DNA. As I told you, not the entire DNA in one strand, only a particular portion. So here unzipping has taken place and forms an open loop once the DNA strand unzips, because only then the information can be copied to messenger RNA. So it will unzip and it uses ribonucleoside triphosphates. There it was, in replication, it was deoxynucleoside triphosphate. Here, using ribonucleoside triphosphate, polymerization process will take place following the rule of complementarity. Why it is said the rule of complementarity? Suppose here it is A, U, G, C. So based on, so the messenger RNA formed of the ribonucleoside triphosphates added also will be complementary. So the nitrogen bases here, it should be uracil, it should be adenine, C, G. So that is also carried out in a complementary or based on complementarity or based on the complementarity of the nitrogen bases. So only those which are matching, only those nitrogen bases which are matching will be present on, will be found on this RNA. Also RNA is formed in this particular pattern. Following the rule of complementarity, polymerization process will be taking place. That is why we are saying the genetic information is getting copied to this RNA. The genetic information present on DNA, the segment of DNA is getting copied to RNA. And this will proceed. The elongation process will take place and only a short stretch, a small portion, a small segment of polymerized RNA remains attached with the enzyme. So here you can see in the figure only a small portion of the polymerized RNA is getting attached. Only a small part is attached to the only a small part is attached to the enzyme. The process of polymerization continues till it reaches the terminator. It has started from promoter and till it is reaching terminator, this polymerization will continue with the help of this RNA polymerase enzyme. And then once it has reached the terminator, again the RNA polymerase needs the assistance of Rho factor or termination factor, and with the help of which it will recognize the terminator gene or the terminator sequence, and once it reaches the terminator, the RNA polymerase and the nascent RNA, means the newly formed RNA, both will fall off. So this process is continued till the terminator, and as soon as it reaches the terminator, both the enzyme, as you can see here, the enzyme as well as the enzyme as well as the newly formed RNA will fall off, will get separated from that area. RNA polymerase gets separated from the DNA and also the transcribed RNA. Same sentence, same thing is mentioned, both of them. RNA polymerase enzyme and the RNA form, the transcribed RNA form will get separated because the process has been completed. Only up to terminator. It will not proceed further. It will start from promoter, proceeds till terminator. As soon as the terminator is reached, with the help of the Rho factor, it can recognize, the enzyme can recognize the termination factor, so it will realize that it is time to stop. So there it will stop and the enzyme will be released from there, and it will again continue the process with other strands, and the newly formed RNA also will get separated from there. That results in the termination of transcription. So I'll repeat the processes once again. The process of transcription is completed in 3 steps: initiation, elongation, termination. These are the three steps. And already you learned about a transcription unit. And you know that the process of transcription begins at the promoter, ends at the terminator. And RNA polymerase is the enzyme. DNA dependent RNA polymerase enzyme is the one which catalyzes the process of transcription. So that will bind with promoter. Only then the process can start. But RNA polymerase, they can only catalyze the process of elongation. They cannot recognize the promoter site. For that, they need the assistance of sigma factor or initiation factor. So they will, or this particular enzyme will bind with, will associate with this sigma factor, and then with the help of it, it will recognize the promoter site and will get attached over there. And the process will start. The process of transcription will start. Then comes elongation. Now binding has taken place. Now the next step is the information present on the DNA should be copied to an RNA molecule, or a new RNA molecule need to be synthesized. So once the RNA polymerase has got attached to the promoter, the unzipping will take place, because without unzipping the message is, because if both of them are coiled together, if the two strands are coiled together, the message cannot be copied to, or the information cannot be copied to RNA. So unzipping takes place, and then slowly a new RNA will be formed using ribonucleoside triphosphates as the substrate. So in the previous video, so when we saw that replication, there I had shown how the new DNA strand is being formed by attaching these deoxyribonucleoside triphosphates one by one. So similarly, these ribonucleoside triphosphates will come and get attached, and they will form a new RNA strand. And this formation of RNA strand is based on complementarity, because there is a sequence of nitrogen bases on DNA. So when a new DNA strand is, a new RNA strand is being formed, that will be based on complementarity. Only matching nitrogen bases will be formed. I hope you understood what is meant by rule of complementarity. So if you compare the DNA strand which has taken part in transcription and the new messenger RNA, or the new RNA formed. See, say for example, if this is the DNA strand, so A, U, C, C, G, U, okay, let it be like this. So this is the DNA strand that took part in transcription. So the messenger RNA formed is based on the complementarity rule. So that means the new strand formed will be having matching or complementary nitrogen bases. Here it will be uracil, because it is, okay, here I wrote, just a minute children. A, T, C, C, G. The new strand, U, A, G, G, C. So the new strand, this is RNA formed, newly formed RNA. This is the DNA strand that took part in the transcription process. So when the new RNA is being formed, that is based on complementarity. You know that always adenine pairs with thymine, cytosine pairs with guanine. But if it is an RNA, thymine will be replaced by uracil. So based on the complementarity, or the rule on rule of complementarity, the new RNA strand will be formed, and only a short stretch of polymerized RNA will be found attached to the enzyme, as you can see in the figure, and it will proceed, the process will be continued till it reaches the terminator. And once it reaches the terminator, termination process, or the process will come to an end. This also cannot be brought about by the enzyme alone. For that, it needs the assistance of termination factor or Rho factor. So they will bind with this enzyme. This Rho factor will associate with this enzyme. Together, they will recognize the terminator, and once the termination has taken place, the newly formed RNA and the enzyme will get separated from there. The transcribed RNA or the newly formed RNA and the enzyme will get separated. Okay, is it clear? This results in the termination of the transcription process. So this you can see a symbolic representation, a symbolic video. Just a minute, you can see what is moving, that is the enzyme. RNA, about the rho factor and the other factor, termination factor is not shown, but just to show you how it is happening. See, enzyme coming and attached, RNA polymerase enzyme, unzipping taking place. And you can see that red color, that is the new RNA being formed. And later, both of them will get separated from there. So what you see is the DNA strand. And once the enzyme or DNA dependent RNA polymerase is coming attached, unzipping takes place, and based on the rule of complementarity, a new RNA strand is formed. Once the process is completed, the RNA strand formed and the polymerase enzyme will get separated from the DNA strand. Any doubts, children? Anything to be repeated? Any point to be repeated?
Transcription process.
Transcription?
Termination.
Termination. Termination will occur when when the enzyme reaches the terminator gene or terminator, because a transcription unit has got three main parts: promoter, structural gene, and terminator. So once it reaches, in the video which I showed just now, you might have seen how it is moving from one end to the other. Starting from promoter, it will be moving towards the terminator. Once it reaches the terminator, with the help of the termination factor or rho factor, it will recognize, this is the terminator. Now it's time to stop. So once it happens, or once it reaches termination, the enzyme will fall off from this because it has got attached to the promoter. So it will get separated from the DNA strand, and the RNA, newly formed RNA, that also will get separated from the DNA segment or DNA strand, and both will be free. And that RNA which is formed, it has to undergo several procedures which you will learn. But the enzyme will again go back in action, that you know. Enzymes, they will help in speeding up a particular reaction. They can be used again and again to continue the process. Again, they can assist DNA segments to carry out transcription. Is it clear? So we were learning about transcription. Transcription means copying genetic information from DNA to RNA. So that you have to be very careful. Sometimes they will give a segment of DNA. The sequence of nitrogen bases in a DNA will be given, and you will be asked to write the sequence of nitrogen bases in the transcribed messenger RNA. So you should remember how is the pairing taking place, and that thymine will not be found in our RNA. Instead, it will be uracil. All that points you should remember, and hope you have understood. And I told you that a transcription unit has got three parts: a promoter, terminator, and also structural gene. And transcription process, it is completed in 3 steps. Which are those 3 steps, Sharon?
Initiation, termination, and
Elongation. Okay, these are the 3 steps. And tell me about initiation, Meenakshi Bijoy.
The RNA polymerase will bind to the promoter and the process of initiation starts.
Yes, how they can bind, or how do they recognize?
This is recognized by the sigma factor initiation.
Yes. So with the help of it will associate, or this RNA polymerase enzyme will associate with this sigma factor, and then they will help in recognizing the promoter, and the process starts. And Punya, elongation?
RNA polymerase uses the ribonucleoside triphosphates of the DNA strand to synthesize the complementary RNA.
Ribonucleoside triphosphates, not of the DNA strand. They will make use of ribonucleoside triphosphates and they will help in elongating based on the rule of
Complementarity.
complementarity. And this will be proceeded. Elongation will be continued till Malavika.
Till the enzyme reaches the terminator gene.
Then what happens once it reaches the terminator gene, Evelyn?
It will be recognizing the termination factor so
With the
that
help of termination factor. And once the termination is done, what will happen? What are the changes followed?
then the RNA will be removed, newly formed RNA, and the enzyme also will be removed.
Okay, will get separated from the DNA strand. Nandana, I saw the doubt in the chat box just now. I'll clear it, clarify next period. Okay. So thank you, children. See you next class. We'll continue transcription process in the next period also. Okay, bye.